This lecture is going to deal with the topics involving protein folding, stability, and protein prediction theory. The topics discussed will be the Chow-Fasman rules, which involve looking at individual amino acids and their propensity to be in a certain secondary structure. The Kite-Doolittle plot, which looks at hydrophobicity and hydrophilicities of individual amino acids. Hydropathy plot talks about the amino acid, again, being involved in its affinity towards water or its repulsion against water. So here we use hydropathy plot really to predict regions of the protein that are going to span the membrane if you're talking about membrane proteins. So it has a lot of utility with regards to membrane proteins, and which regions of the protein are going to be exposed to the outside of the cell or inside of the cell, which is more polar. So hydropathy plot is generally used as a predictive tool for membrane proteins. And we will talk about the Anfinsen experiment. So it's a classic hallmark experiment in biochemistry that deals with protein folding and refolding. And there's the Levinthal Paradox, which really refers to how a protein will fold inside the cell. So folding inside the cell of a protein is dramatically different than folding conditions that are performed in the lab or on the benchtop. So Levinthal Paradox and some of the theories to circumvent the paradox have to do with how a protein folds in vivo or inside the cell versus in vitro, which would be on the lab bench or in a test tube. Finally, we'll discuss the protein folding funnel, which sort of explains the Levinthal paradox. And then there's a brief discussion on chaperones and the proteasomes such as GRO-ES, GRO-EL, which is sort of a garbage can that helps a protein fold inside, sort of sequester it away from the crowded macromolecular environment of the cell. So first thing is the Chow-Fasman rules, which began in 1978. So the goal here is to take a protein's primary amino acid sequence and try to derive its protein's three-dimensional structure. And Chow-Fasman actually looked at secondary structure. So they did a lot of math and a lot of statistics. And what they came up with was a probability or a propensity. So this P stands for probability or propensity, and it's a statistical treatment of proteins and where they would reside in terms of their ability to be in alpha helices or in beta strands. So Chow and Fasman actually worked with only a small subset of proteins, about 13 or 14 proteins whose three-dimensional structure was known at that time. So when I say their three-dimensional structure was solved or known, that is, we can take every atom of that protein and plot it in an XYZ coordinates. So the XYZ coordinates of every atom of every amino acid of every of that protein, all 13 or 14 of them were known either through crystallography or NMR. So using that list, Chow and Fasman developed a statistical treatment. And what they came up with was the ability of amino acids to be, or their predilections to be, in an alpha helix or a beta strand. So if you look at these numbers, there's no units because, again, it's just a probability. It's a statistical treatment of each individual amino acids and their placement in either an alpha helix or a beta sheet based on those 14 or so structures whose three-dimensional structure were known. So if you see here, you have something like glycine, who Chow and Fasman stated has a probability of about 0.57 to be in an alpha helix. Now we know, at least 40 years or so later, glycine is actually enriched in many alpha helices, particularly those that are in membrane proteins. And glycine is also found in a lot of alpha helices that are structural in its functions, such as collagen. So there has been a lot of modifications of the Chow-Fasman rules since. However, the basic premise stays the same. We're looking at amino acids and their ability to reside in either alpha helical secondary structure or beta strand secondary structure. So really they got the ball rolling, Chow and Fasman did. Years later, in about 1982, there was the Kite-Doolittle scale, and this has to do with hydrophobicity. So hydrophobicity means how would that amino acid partition or react with water. So would an amino acid tend to be attracted to water and have its side chains to be exposed? Or would that amino acid repel water and be hydrophobic and sort of do anything in its power to get away from water and its polarity? So given that, Kite and Doolittle developed a hydropathy scale. Here, the higher numbers actually represent the more hydrophobic amino acids on their scale, and the negative values actually represent more of the polar or charged amino acids on their scale. So isoleucine, according to Kite and Doolittle, is the most hydrophobic amino acid. So it would tend to be buried inside a protein, or if it's a membrane protein, probably in a transmembrane region, the sort of hydrophobic interface. And according to their scale, arginine is the most polar amino acid, tend to be more exposed to the exterior or be more exposed to the outside or inside of the cell if it's a membrane protein. So this is a scale, and they have different parameters for their scale. Maybe it has to do with their ability to be partitioned to water. But the key is in the next slide is that not all of these scales are set in stone. So Kite and Doolittle determined based on their experiments that isoleucine and valine were the most hydrophobic, while arginine was the most hydrophilic. So other investigators came up and arrived with different scales. So it depends really on the parameters in which they determined what was most hydrophobic versus what was most hydrophilic. But the scales are not really set in stone. They're not really definitive. So Wolfendien et al., Janin in the late 70s came up with their own versions of hydrophobicity and hydrophilicity, and it's based upon their experimental models and systems. So again, not really set in stone when it's regarded to what is most hydrophobic and what is most hydrophilic. Hydropathy plots, based upon what scale you're using, it could be the Kite-Doolittle plot, which is probably the most famous plot. If you take their numbers that were obtained and sort of plot those numbers assigned to each amino acids, their hydropathy, against their amino acid number in the primary sequence, you get a graph that kind of looks like this. And the hydrophobic regions in the Kite-Doolittle plot are more positive. The hydrophilic regions in the Kite-Doolittle plot are more negative. And what you actually see here is the potential to predict, at least make an educated guess on what regions of a protein tend to be hydrophilic and be exposed to the outside or inside of the cell if it's a membrane protein, or what regions would actually span the membrane, the transmembrane interface, which tends to be more hydrophobic. So if based on prediction alone, you can expect this region or this stretch of amino acids or this stretch of amino acids from residues 200 to 210 or so to span the membrane, and these residues here to sort of be exposed to the outside or inside of the cell where it's mostly polar. So that's the purpose of a hydropathy plot, generally used in membrane proteins, but it can also be used in cytoplasmic proteins as well. Very important in membrane proteins. For example, G protein coupled receptors, which span the membrane 7 times, generally do not have good structural models of membrane proteins because, remember, they have to fold within the context of the phospholipid and the head groups and the chains, the acyl chains. So without a good structural model, all we can do is predict. So if we had a G protein coupled receptor with its hydropathy plot, and looking at maybe the Kite-Doolittle scale or any other scale in the previous slide, any other metric, you can predict which regions would span the membrane and which regions would be outside or inside. Now let's talk about the Anfinsen experiment. It is sort of a hallmark experiment in protein folding science, and it's studied in mostly or universally all introductory biochemistry classes. Before we talk about the Anfinsen experiment, we have to introduce two chemicals. These are known as denaturants. And another term that's given to these chemicals are chaotropics or chaotropic agents. So chaotropic means it causes chaos or it generates chaos. And here are two molecules that can do that. One of them is urea and one of them is guanidium ion. You may remember the guanidium group. It's the side chain of arginine, so this is just a derivative of the side chain of arginine. So these are two chemicals that are often found in the lab, and what they do is, if you expose a folded protein to them, they will cause that protein to denature. So the mechanism behind that, if you see these structures closely, you'll see a lot of the amide groups, and then you see this carbonyl group, C double bond O of urea. What they do is they actually outcompete for hydrogen bonds. So a protein that's folded, one of the tertiary structural determinants that allows or drives the folding are hydrogen bonds. And what you do upon exposure to these denaturants or these chaotropic agents is that they actually outcompete for the native hydrogen bonds and cause the protein to unfold into more of a random type of conformation. So you have chaotropic agents, and then you have agents such as beta-mercaptoethanol, also known as BME, and also known as 2-mercaptoethanol. And so what these chemical agents actually do is that they catalyze a disulfide bond reduction. So you can remember another important tertiary structure determinant is cysteine disulfide bonds. So the addition of BME or mercaptoethanol, what it does is that it converts these disulfide bonds to free thiol groups. And once you have free thiol groups, these become free cysteines, and the disulfide bond has been reduced. So the mercaptoethanol in exchange actually gets the disulfide bond. So mercaptans or beta-mercaptoethanol really catalyzes disulfide bond exchange. So shown here on the left is the oxidized form of the disulfide bond, and shown here on the right is the reduced form with the free thiol groups. The disulfide bond was taken by BME, that agent. So now let's look at the actual Anfinsen experiment. Anfinsen's model protein was RNase A, so that is the protein he did his experiment on. And the function of RNase A is basically to cleave RNA molecules. It cuts up RNA. One thing about the fold of RNase A is that it has 8 cysteines that allow for 4 disulfide bonds. So in this situation, you're going to need to have, when RNase A folds, you're going to need to have the right cysteine get with its right disulfide bonded partner. So the red cysteine must form a disulfide bond with the red cysteine somewhere else in the protein's primary amino acid sequence. If that happens, then you have started a correct fold of that protein. But this red cysteine, if it forms a disulfide bond with this green one, that's an incorrect disulfide bond arrangement, and the protein is not folded properly. So what was done based on statistics alone was that if this red cysteine forms and finds its correct disulfide bonded partner with the other red, then the probability of the blue one finding its correct partner is 1/7. And the probability of the third cysteine to find its correct partner is 1/5. The probability of that second one is 1/3. And the probability of that last cysteine to find its correct disulfide bonded partner is 1:1. If you just do probability alone of it finding its right partner, you would expect 1% of RNase A to be properly folded. What actually happened was that Anfinsen uncovered nearly 100% of RNase A activity was found when he removed these denaturants. So when he added these denaturants and subsequently removed it, probability and statistics actually account for only 1% to be properly folded and to get proper function. He actually found close to 100%, maybe like 98 to 100%. So I usually like to joke that protein folding proved mathematics, or at least in this case proved statistics wrong. So let's look here at the actual pictorial depiction of the Anfinsen experiment. So here you have the native form of the RNase molecule, and there you have the correct disulfide bonded partners. Anfinsen exposed it to 8 molar urea and beta mercapta ethanol. So you have to remember what 8 molar urea does. Okay, it outcompetes for hydrogen bonds and causes the protein to form a more random coil arrangement. So the protein becomes denatured. And the beta mercapta ethanol, what it does is that it reduces all the disulfide bonds. So now you have a whole bunch of SH groups. Now you have a whole bunch of free thiol groups. So what happened next was that he exposed a portion of the RNase A to mercapta ethanol and urea at step 1. But then he started to remove them one by one. So when he first removed only the mercapta ethanol, he found that the protein starts to form disulfide bonds. Okay? But they may not be the right disulfide bonded arrangements. So when he removed the mercapta ethanol, the disulfide bonded arrangements begin to reform, but they may not be the right disulfide bonded partner. When he removed the urea, the protein folded, but he got what's called a scrambled protein. So this is a protein that is folded, but it's not exactly in the right three-dimensional structure so that you have the proper function. So what was next was, after removal of the denaturants, he added a small bit of the mercapta ethanol back. So what the mercapta ethanol did, so you have to remember the mechanism. Mercapta ethanol catalyzes disulfide bonded exchange. So any members of the scrambled protein whose conformation did not have the right disulfide bonded arrangements amongst those 4 cysteine disulfide bonds, the addition of trace amounts of beta mercapta ethanol allowed for the proper disulfide bond to form. And now when you remove the final trace bit of the mercapta ethanol, as well as removal of any of the denaturant, he actually achieved the properly folded protein. How did he know he was able to achieve the properly folded protein? Because he was able to see RNase being cleaved. In fact, he got about 98, close to 100% retrieval of the function. So for all that it's worth, the Anfinsen experiment actually proved one of the central tenets of protein folding science, and that tenet is that the protein's primary amino acid sequence actually determines its tertiary structure. That is to say that the protein's primary sequence, that linear chain of amino acids, has the program to actually allow for the protein to get its native three-dimensional structure. Let's talk about the Levinthal paradox. Here we're talking about a situation that's more akin to the cell, inside the cell, or protein folding in vivo. So this was a paradox that was enunciated by Levinthal, Cyrus Levinthal, approximately in about the 50s or the 60s. And he came up with this thought experiment. The thought experiment was that if you have an amino acid that has 3 possible conformations, and if that amino acid, if that protein is 101 amino acids in length, that means that that protein of 101 amino acids can have potentially 5 times 10 to the 47 configurations, 3 stable states, 101 amino acids for 100 peptide bonds. So those peptide bonds, phi and psi angles, side chain folding, there's potentially 10 to the 47 conformations. Now, does the protein sample all 10 to the 47 configurations and conformations? When we're talking about sampling, we're talking about bond rotations, getting the right hydrophobic interactions, getting the right disulfide bonded arrangements, getting the right phi and psi angles to settle. So if a protein samples all 10 to the 47 configurations every 10 to the 13 seconds, Levinthal found that it took 10 to the 27 years to find the right configuration. That is, it took several hundred, if not thousands, of centuries. Real proteins inside the cell do not have centuries or thousands of centuries for it to fold from milliseconds to microseconds. In actuality, it takes anywhere from milliseconds to microseconds for a protein inside the cell to find its correct native three-dimensional structure and hence achieve its function. So something is really happening here that sort of circumvents the Levinthal paradox inside the cell. One of his conclusions was that the cell just simply cannot experiment and explore all possible configurations before it settles on one stable state. So to sort of answer this paradox, there's two explanations. One of the explanations is that the cell has chaperones, which are proteins that help the other misfolded protein get to its right three-dimensional structure and shape. The other one has to do with a more thermodynamic treatment of protein folding, and that is the protein folding funnel. So in the protein folding funnel theory, a protein undergoes several conformations. It has several conformations that it starts out with, and then it experiments with several subsets. So instead of going through every possible dihedral angle and every possible rotations of bonds before it finds their correct native form, what it does is that it explores a subset, and then it sort of undergoes local energy maximas. So here you have local energy maximas before it settles on a local energy minima. So a local energy maxima may be where a bond or stretch of amino acids are changed or rotated before it settles and gets into a minima. Or maybe a energy from the system is inputted so that a domain or a stretch of amino acids rotate, and then that rotation or that conformation allows for a more thermodynamically energetically minima here. So what happens is that the protein in its folding pathway pays a small price in terms of bond rotations and dihedral angle exploration to achieve a thermodynamically lower price. Okay, and then it changes again a stretch of amino acids or domains before it allows it to go to an even lower price enthalpically and entropically before it finally settles on a native protein fold. So shown here on the right is one theoretical model. And one thing is that you have this back and forth. This is an equilibrium. So this is totally unfolded, and then it finds a fold, and then well, this satisfies the thermodynamic criteria, so then it can go to the next state, and it can graduate to the next state. But maybe this is not so thermodynamically feasible. This is not the right fold or the right arrangement, so it goes back, and it fine-tunes and gets another conformation. So like that, this back and forth, sort of this permutation occurs inside this folding funnel before it settles on the correct native state. Couple of things about the folding funnel. Number one is that you have several different conformations. Okay, you have several different forms of the protein that gets initiates this funnel. It's not one conformation settles on one folded structure. It's several different conformations that settle on one folded structure. And the second difference or deviation is that you have several local transition states, and you have several local activation energy barriers before it finds it settles on the right state. So this protein folding pathway sort of answers one question of why it takes seconds to milliseconds, some cases picoseconds and microseconds, for a protein to find its correct structure, and very importantly, because of which it has a correct function. So the folding funnel diagram is actually three-dimensional. So here the protein can start off in conformation B, go through some humps, enthalpically speaking and entropically speaking, before it achieves a minima. Or it could start at conformation A, and that's a lucky conformation because from that conformation it folds all the way down to the native state. Some cases it's just more complicated. The protein experiences many different conformations and degrees of freedom before it settles. So it really doesn't experience all the conformations, it just takes too long, but it experiences a subset of conformations from several different intermediaries that are energetically high and then energetically low, local minimas and local maximas, before it just falls down nicely into the right form. It just sort of collapses to the native state. So that's one explanation to sort of circumvent the Levinthal paradox. Let's talk a little bit about chaperones. So our cells actually contain protein disulfide isomerase and a whole series of other proteins whose main job is to actually help other proteins fold properly. So the cell dedicates a whole series of what are known as chaperones, and these chaperones or chaperonins take a protein that's misfolded and give it a scaffold or a platform for it to find its native three-dimensional structure. So the Anfinsen experiment is very interesting in this regard. So if we go back to the Anfinsen experiment with RNase A, Christian Anfinsen actually found RNase A to fold properly and renature to close to 100% of its activity within a time span of 10 hours. Okay. Now, our cells don't have 10 hours to get a protein to fold and renature into its proper form and hence achieve its proper function. But our cells do have protein disulfide isomerase. So very interestingly enough, if you take unfolded RNase A, remove any denaturants, but put in PDI, or protein disulfide isomerase, you'll see that RNase A refolds in a time span of not 10 hours, but in a time span of about 4 minutes, 2 to 4 minutes in fact. So these cells that contain these chaperonins dramatically reduce the time it takes for a protein to get to the right fold. So mechanistically speaking, PDI catalyzes disulfide bonded exchange. This is an enzyme found in our cell, and it probably helps facilitate the protein to fold properly, as all chaperonins do, but it probably also lessens the impact of the Levinthal paradox so that you don't have this ridiculous time it takes for a protein to find its 3D form. And here is your disulfide bond exchange. So here's the improper fold of the protein. You can imagine this being targeted by PDI. You have this mixed disulfide. This other stretch of the protein can find the correct disulfide bonded folding arrangements. And now, as you see on the right, the correct fold as opposed to the improper fold here that's shown on the left. So there's this sort of ebb and flow, a give and take between free thiols and oxidized disulfide bonds, the back and forth experimenting, reformation, and the breakage of disulfide bonds till you get the right one, and PDI helps in that process. Another class that's not shown here are the heat shock proteins, HSPs. So that's just a whole series of proteins whose job is to help other proteins fold when they get denatured. Usually these heat shock proteins get activated when there's high heat or high thermal elevation inside the cell. The GROEL, GROES. This is essentially a big garbage can with a lid. And inside this garbage can, you have a very nice secluded area for a misfolded protein to get into the proper environment for it to experience and experiment proper folding. So you have a GROES lid that has sort of a 10 angstrom hole, and this whole thing is 140 angstroms in diameter, this sort of garbage can with this lid. And the garbage can lid, the GROES, opens, and then the protein can go in and it is sort of secluded from the macromolecular crowded environment. This is very much an ATP dependent process. The cell dedicates a tremendous amount of subunits, each with their own rotational symmetry, and it dedicates a lot of other proteins whose main job is to target misfolded proteins and allow them to fold. This thing has a height of 184 angstroms, so you can actually see these complexes under an electron microscope. They are very big. So there's actually a mechanism for which this takes place that is shown in the lecture slides. You do not have to know that mechanism, but it's good to just peruse over. One of the things you'll find is that this is highly an ATP-dependent process, about 7 or so ATPs. Another thing you'll find is that a protein can go through multiple iterations, and it may come back a better folded protein so that it can do the job by itself in the cytoplasm to become fully folded. And another thing you'll find is that this can do two. Okay, so one folded protein can go here and properly fold, and another misfolded protein can go here and properly fold. There's also a lot of conformational changes. This lid actually switches and rotates 180 degrees, again requiring the process of ATP hydrolysis for that to occur. This is not a 100% fail-safe mechanisms. Proteins still misfold, proteins still aggregate, proteins still become mutated. And as a result, we can have several terrible diseases as a result. More specifically, when a protein aggregates, you get a lot of neurodegenerative diseases, but also the protein can just simply misfold, and you can have diseases coming from that misfolding as well. So the conclusions of this talk is, number one, the Chow-Fasman rule sort of kick-started this whole process of prediction, using primary amino acid sequence to look at and predict tertiary structure and three-dimensional folds. And Chow and Fasman looked at it at the level of a secondary structure and the propensity or the probability of amino acids to either be in alpha helices or be in beta strands, very limited in the number of proteins they looked at to base their statistics off of. The Kite Doolittle plot looked at amino acids and their partitioning with water. They came up with a scale of the most hydrophobic and the most hydrophilic amino acid. Other scales come up with other amino acids that are most hydrophobic and most hydrophilic. So again, this scale is not set in stone. Hydropathy plot is greatly used to predict structures whose proteins whose structures have not been solved. And it can predict, particularly for membrane proteins, which regions would span the membrane and be hydrophobic. The Anfinsen experiment looked mostly at the denaturation and subsequent renaturation of RNase A. The Levinthal Paradox talks about just the sheer amount of time it takes for a protein to explore all regions of conformational space in order to find the correct fold. It takes several centuries. It can't happen inside the cell in vivo. So several explanations for this. One is the protein folding funnel, but several local activation energies, several local transition states before it collapses to the folded form. The cell dedicates chaperones such as HSPs, heat shock proteins, and as we've shown in a slide here, PDI, protein disulfide isomerases, help the protein fold, help the protein get its proper disulfide bond arrangements, and that could sort of help explain the Levinthal paradox as well as the protein folding funnel. And then finally, we discussed the proteasome. Proteasome is a tremendous complex that sort of sequesters or hides the protein, gives it its own space, gives it its own environment for it to properly fold. You can imagine inside that sort of chamber of the proteasome, GROES, GROEL, the protein is experimenting and experiencing several conformational changes, several rotations, and several domain shifts before it settles on one that is its properly folded form. The GROES, GROEL that you've seen in our slides is actually from E. coli. Eukaryotes have their own version of the proteasome. It has much the same way, has the shape and form of GROES and GROEL.